Hello, my name is Bianca Brattoli, and I'm a junior at Fairleigh Dickinson.
Hello, my name's Trishauna Leah, and I'm a junior at Fairleigh Dickinson.
Our research that we have been currently conducting has been the characterization and binding studies of catalytic domain of MMP9. So, a little bit about our research. We studied the MMPs, which are made up of zinc and have a calcium dependent and are calcium dependent, which are known to degrade elements of the extracellular matrices. They are subtly regulated to carry out various physiological functions like extracellular remodeling, wound healing, angiogenesis, and etc. Overexpression of MMP9 may occur in various pathological conditions, just like hypertension, and we are studying the binding of the natural catechol derivatives to an active form of the MMP9 created synthetically by expressing in E. coli. Here we are trying to research and study how natural inhibitors called tissue inhibitor of metal proteinases, TIMP, bind to our catalytic domain of MMP9 and MMP1. Some background information. MMPs are crucial and important to clinical research due to its involvement in remodeling of the extracellular matrix. Research has been done to identify what would cause MMPs to be inhibited when treating or preventing diseases such as cancer. Um, our matrix metal proteinases are proteinases belong to a family of endopeptidases, peptides containing 23 members. So there are 23 MMPs that can be found in the body, but the for our research purposes, we are only looking at MMP9 and MMP1. Um, our objective that has been involved in our research has been to characterize our synthetic MMP9. We have been studying the conformational changes of MMP9 with various modulators like dopamine and epinephrine, testing the effect of TIMP on the MMPs with and without exogenous modulators. Some methods that we have used in the lab have been taking MMP9 enzyme and diluting the solution. We have made epinephrine stock solution, which was created in the lab. And we also have taken before MMP9 samples with and without epinephrine and with and without TIMP. They were added to a 96 well plate and incubated in a plate reader for 45 minutes to an hour. For the MMP9 experiment, we always use 10 microliters of substrate, and that was also added to the plate after incubation. And these experiments were done in triplicates.
Okay, so our this is a sequence of our synthesized MMP9. It shows you the how what amino acids are involved in the synthetic MMP9. We also ran a gel to confirm the location of our enzyme, which was found to be around 35 kilodaltons, and the Western blot was done to further confirm that, and it also showed up at around 35 kilodaltons. So we did a CD spectra of various MMP9 samples to find out which modulator would cause the MMP9 to change its conformation. So the lowest line on the CD spectra is just the regular MMP9 enzyme, which is our standard, and we used epinephrine as our modulator, which we found had like a conformational change to the protein, which is the reddish pink line above. So our graphs show that for the synthetic MMP9 and TIMP, when added to the sample, there was a decline in activity with just the enzyme, the substrate, and the TIMP. But once epinephrine was added, it declined further than just TIMP, as the epinephrine and would bind to where the substrate probably is, and that would cause like the TIMP to bind somewhere else and the substrate to not really have anywhere else to bind, which would cause a decrease in activity. And the same thing was done for the MMP1, which showed that the enzyme plus epinephrine and TIMP would cause a significant decrease in activity. So for our conclusions, we found that when TIMP was added to the MMP9 along with the epinephrine, the activity of the enzyme began to decline significantly compared to just the MMP9 with shrimp and with substrate and TIMP. This reiterated the fact that tissue inhibitors of metalloproteases bind better to synthetic MMP9 in the presence of epinephrine. And epinephrine is a known modulator in our lab, which might be associated with the conformation change of the protein. There was a similar inhibition with MMP1 and TIMP in the presence of epinephrine. And from our little diagram below, that was just to confirm that epinephrine was supported in the conformational change of the protein, as when epinephrine was involved with TIMP, the activity of the substrate leaned more towards the E prime aspect. So our MMP9 was synthesized by GenScript USA, and our MMP9 spectra was done at the Keck Biophysics facility at Northwestern University, and we also had a grant for this experiment.